Hi everyone, my name is Jasmine and I'm a master's student in the animal science department, and today I'm going to talk about whole genome duplication events and their effects on evolution. So a whole genome duplication event is exactly as it sounds. It is when the entire genome is duplicated and the resulting duplicated genes from these events are called onologs. Now, these events are known to be a dominant factor in improving species diversity and evolution. However, little is known about how this evolution occurs because we are limited in studying this process, as most events are considered too ancient. Luckily, we are able to use rainbow trout as a model, as they have gone through two more recent whole genome duplication events, one 320 million years ago and one 80 million years ago. And it is this most recent event, referred to as SS4R, that allows us to study evolution as it's happening. Now, when choosing how to assess evolution following whole genome duplication, we chose to look at a cell signaling pathway, specifically the activin receptor signaling pathway. Pictured here is a human protein interaction network of the genes associated with this pathway. And as you can see, it is very complex. But in rainbow trout, this pathway is exponentially more complex, as rainbow trout have retained 2 to 4 onologs of each gene. And this complexity is exactly why we chose to use a cell signaling pathway, as under whole genome duplication scenarios, entire gene networks are simultaneously undergoing functional evolution, and therefore changes in one member will directly affect another. In order to study whole genome duplication in this pathway, we collected RNA from tissues chosen as a representation of the whole body and applied nanostring technology to get exact numeric expression counts for just our genes of interest. And once we had this data, we wanted to understand the evolutionary differences between these genes. And so we chose to first look at differences in tissue specificity across both individual genes and then when comparing molecule type groups. From this, we found that inhibitors were the most tissue specific and signaling proteins were the least, and that these two groups were only significantly different from each other. In addition to comparing the expression across molecule types, we wanted to assess how the onologs were evolving in relation to each other. So when looking at the TS3R onologs, there is no clear pattern within the onologs themselves, but there is a pattern across tissues. Now, this could show that over time, the tissues have specialized to one set of onologs over the other, such that the receptor, ligand, and signaling protein are all from the same origin. In direct contrast, when we look at the SS4R onologs, there's a very clear dominant form for most genes. Now, we're still working through some of this data, and we are actually starting a new project to see how manual manipulation of evolution may cause changes in expression of related onologs. Essentially, if we knock out the function of one gene, how are its other onologs affected? To do this, we are using CRISPR-Cas9 to knock out each of the 4 activin A onologs, and we will apply nanostring to samples taken from these individuals as well. So with that, I would like to thank you for your time.